So my name's Dr. Emma Rocke. I'm a marine microbiologist here at UCT. We are particularly interested in understanding red tides and their interaction with the microbiomes. A red tide is a phytoplankton bloom which are red-orange in color. A microbiome is anything in the water that we can't see with the naked eye. All right, so it's important to understand that the microbes, or those blooms, are responsible for producing oxygen. It's really important that those nutrients, as they die, those nutrients are regenerated so they can continue to bloom. And the microbiome is 100% responsible for regenerating those good nutrients. So we can see that as a good microbiome. With climate change, we can get a situation whereby we get an unhealthy microbiome. All of the oxygen is pulled out of the water. These microbes will produce nitrous oxide, which is a noxious greenhouse gas. Under normal circumstances, this gas will stay at depth, and it's a bit like you have a glass of fizzy water or a glass of Coke. If it's cold, the gas, it will stay fizzy much longer. If you start to warm it up, that gas starts to bubble to the surface. So what the concern is our ocean is warming. And if our ocean is warming, these gases that normally stay at depth will bubble to the surface. In order to look ahead and attempt to mitigate something, one needs to understand the source of the problem. And once we understand what is causing these shifts, then we can change activities that are perhaps contributing to that. So we've been collecting some water from the beach. And once we collect the water, it's important that we get all the bacteria and viruses associated with it. We do not want any of the bigger stuff. We want to know how rich the water is in nutrients at this time. Nutrients is what fuels these blooms. So this species is a diatom, which aren't known to bioluminesce. Dinoflagellates are. So bioluminescence is when phytoplankton blooms in low light conditions look like they give off a sort of bluish glow. After we've taken our sample and filtered that sample, that filter is then broken up into tiny little pieces. We then break open the cells that are present on that filter so that we're able to expose and isolate the DNA. The DNA is then sequenced using the MinION in our lab. This is easy to use and it's much cheaper than other methods. It tells us who is in the sample, what genes are present, and more importantly, what genes are active. So once we've collected all of that data, we're able to put it onto a database.
So what does the future of this research look like?
I would love to see in 20 to 30 years a monitoring system whereby we can tell when that microbiome switches over. Where is the tipping point? And so we're able to react accordingly.